Hi everyone, so today we will be discussing DNA replication and its models, proposed models, right? So as you know, DNA replication helps in doubling the amount of DNA that we have in our system, and this step, if you look at the cell cycle, so from M phase, the cells that mature, that are produced, so the daughter cells that are produced, they enter a phase or a cycle which is known as the interphase of the cell cycle. In the interphase of the cell cycle, the cells undergo three stages: the G1, the S, and the G0, G2. In S, which is the synthesis phase, every cell replicates its DNA. Replication of DNA takes place in this step. So replication of DNA basically means to make a copy, right? So if you remember what the structure of DNA is like, it is basically nucleotide sequences, right? Like this on both sides, obviously, and this is the phosphodiester bond that is formed. So now what happens is that in this, the nucleotides are synthesized in the body. What your body has to do is make exact copies of the adenine, guanine, thymine and cytosine bases by reading the template strand. Okay, so there were three proposed models, three basic concepts like ideas through which replication could have happened. So these three proposed models were the conservative model, the semi-conservative model and the dispersive model. Let's start with the conservative model. So the conservative model, so conservation. So the term states what it means. Conservation means to keep something intact or as it is. So in conservative model of DNA replication, you have parent DNA, which as a whole, the entire DNA, entire double-stranded DNA will serve as the template. So it will be the template which will stay as it is, and a new double-stranded is formed. Correct? Again, so in the first generation, then you will have the exact parental DNA. It will not be changed in any way, right? So that is the conservation, conserved parental DNA. And then you would have a new dsDNA. Again, in the second generation, when this thing will replicate again, you will again get conservation of the parental DNA as it is. This is again conservation and a new strand. Here, because it was already the new strand, so again there would be a conservation and there would be the formation of a new strand. So conservative DNA replication says that the entire double-stranded DNA, which is the parent DNA, is conserved, and the new DNA is a dsDNA that is entirely newly forged. Okay, now for semi-conservative, the idea separates. The idea is different. Here they say that semi means half. So here half conservation will happen. What do I mean by half conservation? So here it says that the dsDNA denatures and each strand serves as a template DNA, right? So this strand will give rise to one new set. So one parental, one would be parental, and one would be the daughter of the new strand. Similarly here, one would be parental and one would be the daughter. Again, in the second generation, it's going to be the same. So the parental strand, original parental strand, would give rise to one daughter strand, and the newly formed daughter strand will give rise to its daughter strand, right? So what is noticeable? The noticeable difference between these two is that after the second generation of replication, in conservative model, you will still have a double-stranded DNA identical to parent DNA, right? But here, after the second generation, you will either have hybrids, right? So one strand of parental and one strand of daughter, or you will have entirely new dsDNAs. Correct? So this is the most important thing that is noticeable between the two, that the parental DNA is not conserved. You get hybrids in a semi-conservative model. However, in a conservative model, you will never get hybrid DNAs. Coming to the dispersive mode, in the dispersive mode, it says that the new DNAs formed are basically amalgamations, sections of the parental DNA and daughter DNA forged or mixed together, right? When further experimentation was done, which is known as the Messelson Stahl experiment, it was proved that semi-conservative method is the method that is actually at action when DNA replication takes place in humans. When we looked at DNA replication, we know that because the human genome is linear and large, it gets replicated in sections. So the section of DNA which undergoes replication opens up and forms something which is known as the replication fork. At the replication fork, two new strands are synthesized. However, the 3' to 5' running strand and the 5' to 3' running strands are replicated slightly differently because the polymerase or the enzyme which takes part in replication, which helps in replication, we call it DNA Pol 3, DNA polymerase 3. It is only capable of synthesizing a new DNA from the 5 to 3 direction. So it makes a DNA from the 5 to 3 direction. So then the 3 to 5 strand gets a continuous manufacture, which is known as the continuous strand. But the 5 to 3, the 5' open strand gets a discontinuous manufacture and it's known as the discontinuous or the lagging strand. The sections of DNA or segments of DNA that are formed, they are known as the Okazaki fragments. Okazaki fragments. We will discuss the Messelson-Stahl experiment in detail so as to understand how semi-conservative model was proved, and then we will go on to understand how actually the process of replication takes place. Let's look at Messelson-Stahl's experiment. Okay, so Messelson-Stahl's experiment is based on the concept that DNA is primarily made up of nitrogenous bases, right? So DNA has nitrogenous bases. In every nucleotide, you have either adenine, thymine, guanine, or cytosine. Therefore, during the manufacture of DNA, nitrogen is a primary component that is taken up by any cell. E. coli was selected, Escherichia coli was selected as the model because it has a very quick rate of replication of 20 minutes, right? So because it divides every 20 minutes, its cell replicates its DNA every 20 minutes. What the scientists did was that they initially, for multiple generations, cultured E. coli in a heavy isotope of nitrogen, which is N15, such that the DNA was completely comprising of, so the nitrogenous bases in the DNA completely comprised of heavy nitrogen. When they took this kind of DNA, now because the DNA is same, it doesn't matter, when they took it and did an analytical centrifuge or a density gradient centrifugation with the help of cesium chloride, they found that a heavy band was formed during centrifugation. When this band was photographed using autoradiography, they got a peak which was the N15 peak and a band for N15 as well. Then they took a part of this culture, a section of it, and started growing it in a N14 culture, which is the normal isotope or the lighter isotope. After 20 minutes, that is after one round of replication was done, if replication was conservative, then you would get, this is the heavy nitrogen N15, N15 double strand, and a new N14, N14 double strand, which has been synthesized in the next 20 minutes. If it is semi-conservative, then you would get a N15, N14 hybrid, because they would separate and each would give rise to a new DNA, and because the nitrogen present in the medium is N14, the new DNA will be made with N14, right? So therefore, if we did a centrifugation of this particular, of the sample, for semi-conservative, the bands that we should ideally get is a heavy band and a light band, right? But when the centrifugation was done, they found neither a heavy nor a light band. They found an intermediate band, which when photographed gave us a N15, N14 peak and a N15, N14 band. This was the primary proof that obviously you do not get a heavy band, so there was no conservation. So this proved that there is no conservation of DNA. They repeated the experiment for this next 20 minutes. So in cycle 2 again, when they did it, they found that there were both intermediate as well as light bands. So these were N14, N14 bands, and these were N15, N14 bands, hybrid bands. Again, if it had been semi-conservative, this should have been the bands that we would have gotten, but we did not get it. We got instead a heavy and an intermediate band in the graph as well as the photograph, right? Gradually, when they did it for another 20 minutes in cycle 3, they saw that the amount of intermediate band was lesser as compared to the normal isotope bands. Why? Because again, because replication is semi-conservative, gradually this band will only stay 2 in number, right, into 2, whereas the newly formed bands will keep on multiplying, because every time the original DNA will get split and you would get 2 new DNAs which would be formed entirely of N14, and therefore the quantity of light DNA will gradually increase as compared to intermediate DNA, and there will be never bands found for heavy DNA isotopes, right? This proved, this experiment, which was a very basic experiment, it proved that conservation or conservative model of DNA replication is not right. DNA is replicated by a semi-conservative model. I hope this was clear to you. You get a lot of questions from this in boards as well as in NEETs. You get direct questions as to explain the experiment. You get diagrammatic questions which can be a bit tricky. We will try to solve those questions in the coming videos. I hope this was helpful. Thank you.